रेकॉर्ड होईल का लेक्चर? परवाचे 2 केलं का मॅडमचं रेकॉर्ड? हा, मग ते जरा मला आणि गुजर मॅडम द्या. मला स्क्रीनवर पीपीटी दिसेल ना, या स्क्रीनवर? ह्याच्यावर कॉपी केलंय का सर लेक्चर? आणि हे कुठे पेनड्राईव्ह तुमच्याकडे?
Good afternoon. On behalf of Department of Botany, Telangana University, I wholeheartedly welcome the speaker, resource persons of afternoon post-lesson session. Now I would like to request Professor Aruna ma'am to introduce our speaker, Dr. Yogesh Choche from National Center for Cell Science. Over to Aruna ma'am. Ma'am.
Good afternoon, sir. Good afternoon. Am I audible?
Yes, you are clear, audible.
Afternoon to all the dignitaries on the platform and delegates attending the webinar today. Hope that all of you had your lunch and will actively participate this session where we have got 2 lectures delivered by eminent scientists. And now, myself, Dr. Aruna, Chairperson, Board of Studies of Botany Department, deem it as a privilege to introduce Professor Dr. Yogesh Choche, who is Emeritus Scientist from National Center for Cell Science, and he's also serving as Principal Investigator, National Center for Microbial Resources, Pune. And Professor Dr. Yogesh Choche has started his career as scientist at National Center for Cell Sciences, Pune, and after working on different aspects of microbial, especially bacterial diversity of various ecological likes, his thrust area of research is now to understand succession of microbial communities in human gut during development and their role in health and diseases. And he's working since 25 years in the field of microbial ecology, microbial molecular taxonomy, and biodiversity. And he has published more than 200 publications in journals of relevant areas, reviewed publications in international journals like FEMS Ecology, International Journal of Systematic and Evolutionary Microbiology, and Microbial Ecology. And he's also on editorial board of prestigious journals like Current Science and PLOS ONE. And to all the students, my dear students, we are very happy to hear to a scientist who is serving as a person that is a member of editorial board of prestigious journals, where we will be always discussing to you all in the class, saying that you should have a publication in well-versed and notified and noteworthy
Ma'am, unmute yourself, ma'am. Aruna ma'am.
to hear?
Yeah, now you are audible.
All these was waste?
No, you.
Okay, in 2009, he was given a responsibility of establishment of microbial culture collection. It's now known as National Center for Microbial Resources. Currently holds more than 150,000 bacteria and fungi in its collection, recognized by WFCC, that's World Federation of Culture Collection, and MCC is also recognized as International Depository Authority and is designated repository by Environment, that's Ministry of Environment, Forests and Climate Change, Government of India under the Biological Diversity Act 2002. And in September 2018, Department of Biotechnology extended the mandate of this repository to include antimicrobial resistant microbes. So with these few words, now I welcome and I request Dr. Yogesh Oche to deliver his talk on topic diagnostic tests for COVID detection. And I thank Haleem Khan sir for giving me this opportunity. Over to you, sir, Dr. Yogesh Oche, sir.
Thank you very much, madam. And I hope I am audible and my is my visible?
Yes, yes.
Can you see the presentation now? Yeah.
Yes, yes.
Okay. And is the slide changing?
Slide is not changing, sir.
Yeah.
Your cursor is moving. Your pointer is moving.
Yeah, I know.
Your pointer is moving, sir.
Yeah. Now it has changed?
Yes, yes.
Okay, fine. So, yeah. So, thank you very much for this opportunity and nice words of introduction by Dr. Aruna and Dr. Nandedkar and Dr. Haleem Khan for giving me the opportunity. So, it's a great pleasure to be here and make this presentation. Of course, I'm a little old-fashioned and I would like to have a personal presentation rather than online because audience response is perceived better in a personal presentation than online. But in the present situation, this is the best that we can do. So it's a pleasure for me to make this presentation. And what I'm going to talk about is currently available diagnostic methods for the diagnosis of COVID infection. And to take a second for the slide to change, I suppose. Yeah. So this virus, as we all know, it was first reported in China in December 2009, and then it spread all over the globe, causing what we know as a pandemic today. The advantage is that today, because of the advancement of several technologies, fortunately, specific diagnostic tests could be developed for the detection of this virus immediately. We will talk about that. So although Dr. Nishikantha Naik must have told you everything about the coronavirus in her talk, just to recapitulate some of the things, because maybe during the lunch you have forgotten few things. So just certain things, that this virus is typically made up of a membrane, a protein outer layer, and a nucleic acid. And there are several proteins present in the virus, and its nucleic acid is ribosomal RNA. I mean, its nucleic acid is ribonucleic acid and not deoxynucleic acid. So it has a RNA as a genetic material and not DNA as a genetic material, and that makes a lot of difference in many ways, which we shall see later. But it makes a lot of difference for the purpose of diagnosis also. So these are all coronaviruses are large envelope and single-stranded RNA viruses, and it belongs to a group of related RNA viruses that cause diseases in mammals and birds. And in human and birds, they cause respiratory infections, and many viruses, as we know, H1N1, SARS, all these viruses belong to this category. And why they are called as coronaviruses is because the appearance of spike protein on the membrane looks like a corona. And this is what I said, that outer membrane layer, proteins, and the genetic material, which in this case is RNA. And here you can see the corona-like structure, giving the name coronavirus. So this is basically a spike protein. So when we want to diagnose or detect the presence of virus, there are many methods of testing are available. One test is molecular testing, where we detect the presence of viral genetic material, that is RNA. Secondly, we detect the presence of antigen, that is a protein that is produced by a virus when it causes infection. And third test is antibody test, where we look at the antibody produced in the infected individual in response to virus infection. So presence of antibody is indicative of infection. In analogy, I can give you an example that some of the students who are familiar with medical microbiology, they must be aware of Vidal test, which is done for the diagnosis of typhoid. And in the Vidal test, also Vidal test detects the presence of Salmonella typhi specific antibodies in the infected patient. So this is similarly, presence of antibodies is an indicative of infection. But today for coronavirus, antibody is not used as a diagnostic test. It is used for serosurveillance. So we will not talk much about that. We will spend more time on the other two tests. So since I have already said that virus has a membrane and a proteinous outer layer and the nucleic acid, so I have already told you that there are both can be detected by current diagnostic method. Nucleic acid, viral RNA can be detected by molecular methods, and viral proteins can be detected by using the antibody methods. So you can either detect for the presence of viral specific RNA, or you can detect for the presence of virus specific protein. So that brings me to the point that what do you mean by diagnosis? By diagnosis, you mean that you want to identify the cause of infection accurately. And so your diagnostic test should be very accurate, and it should specifically detect the infection by a particular virus. So it should not give you false positive, in the sense that the person is not suffering from the disease, the said viral infection, but still the test comes positive. That should not happen. And at the same time, it should not also give you a false negative. That is, a person actually has the infection, but the test doesn't come out to be positive. So a good diagnostic test avoids both these scenarios and always gives you a positive test whenever there is infection. So that is something which is important to remember here, that the test should be specific, and the test should be it should specifically detect the virus, and it should be sensitive also. So it is good that if the test comes positive in smallest number of viruses present in the sample. So smaller the number of viruses the test can detect, more sensitive is the test. And smaller, the advantage of smaller number is that then in that case, a more sensitive method can detect the infection at earlier stages. At the later stages, when the viral load in the sample is very high, there is nothing great in detecting at that stage. I mean, many tests detect at that level. But if a test detects presence of virus when the sample, the viral load is low, that is, person has not yet developed active infection, then that test is considered to be better and more sensitive. So this is something which you should remember in this context. So the most popular test used for the detection of viral RNA is RT-PCR. Now, RT-PCR, as you know, it's a PCR, that is polymerase chain reaction, and RT actually has two meanings: reverse transcriptase PCR and real-time PCR. Here it is both. Why reverse transcriptase PCR is, as some of you must be aware, the PCR or polymerase chain reaction is the technology which is developed for DNA, that is deoxyribonucleic acid, and it is a very well established procedure by which DNA can be amplified several million fold in the laboratory. So if you do 30 cycles, then from a single DNA molecule in 30 cycles, you can generate 2 raised to 30, that many number of DNA fragments. So in the first cycle, 1 DNA fragment becomes 2, in the second cycle 2 become 4, next cycle 4 becomes 16, so on and so forth. So that way there is a exponential amplification of template DNA. But as I said, that genetic material for this virus is RNA or ribonucleic acid. And how do we amplify? Then ribonucleic acid using the test which is meant for DNA or deoxyribonucleic acid. So the answer to that was found out by the use of another enzyme which is called as reverse transcriptase, which converts RNA into its complementary DNA. And then this DNA can subsequently be amplified using polymerase chain reaction. So since the test makes use of reverse transcriptase in the first stage, it is called as RT-PCR or reverse transcriptase PCR. And then in a typical PCR, you get the amplification at the end, and typically what you do is that after your 30 cycles are over, you run a gel and look for the presence of a DNA fragment of a specific length. So that is a standard PCR. But over the years, the technology has evolved by which there is a monitoring of the amplification as amplification is taking place in the reaction tube. So there are different technologies, typically by monitoring the amount of double-stranded DNA that is formed. I said that one DNA molecule becomes 2, 2 becomes 4, 4 becomes 16. So as this amplification takes place, the new double-stranded DNA that is formed, that DNA is quantified, and the rate at which the DNA is growing inside the tube is monitored, and then that can be used to find out the initial concentration of DNA. Obviously, if the initial concentration of DNA is high, the amplification will be at the faster rate. Initial concentration of DNA is low, the amplification will proceed at a lower rate, and based on that rate, you can calculate the initial concentration of DNA. And that in turn becomes a very sensitive method to detect the lowest possible amount of the target DNA, in this case, the diagnosis of COVID virus. So specific detection of COVID virus specific RNA can be done using this methodology. So typically the test would involve viral lysis, purification of RNA, then reverse transcription, and followed by quantitative amplification of the RNA, the cDNA. And now anybody will ask a question, where does the specificity comes from? How does this test specifically determines coronavirus or COVID-19? So that specificity comes from the primers that are used for the amplification. The primers are specific for one of the several proteins in coronavirus. So coronavirus has many proteins. I'll show you later. It has spike protein, nucleoprotein, ribosomal dependent RNA polymerase. So it has many such proteins, and the tests are designed that determine, the primers are designed to pick up any of these protein, one or more of these proteins specifically, and amplify that. So the specificity of the test comes from the primers used for PCR. And in simple words, there is a large number of commercial tests available today. All tests differ in the primer which they use for PCR amplification. So this part I will just.
Statuses कैसे आएगा? क्योंकि I think JV doesn't have ODM.
Hello.
And I think.
Hello. Is somebody asking a question? Hello.
So I think Reggie for API you have suggested.
No sir, actually.
Service center होता है वो कैसे से यहाँ पे.
Karla madam, your mic is on actually.
We were saying something else.
There is sound from your mic. Yeah, sir. Now you can continue.
Okay, fine. Because I thought somebody is asking a question. Anyway, yeah. So, okay. So this part I have explained you earlier, but I'll just go through. This presentation will make it more clear. So I said that PCR is meant for DNA. So first you have a single molecule of DNA. During the PCR, that molecule, the specific primers amplify a specific part of the DNA. So these primers are specific for a particular segment of DNA. And the stretch of DNA between two primers gets amplified. So this is one primer, this is second primer, and these primers are the one which bring in the element of specificity. These primers are specific for particular gene or particular fragment of DNA, and the PCR, as we go through multiple cycles of PCR, the region of DNA that is present between these two primers, that is this region, that is a region that gets amplified several million times. So this is a thing which you should remember. And what we do in reverse transcriptase PCR is that from the original viral RNA, that RNA gets converted to DNA first, and then in the DNA form, it gets amplified. And these primers, which are specific for a particular gene of coronavirus, that help us in the specific detection of coronavirus. So the most important part of the diagnostic test is the selection of proper primers. These primers bring out the specificity to your diagnostic test. If you choose right primers, then those primers will only amplify specific coronavirus and not any other related virus. So what you need for the polymerase chain reaction is a specific instrument which keeps on altering the temperatures that are required for PCR. So amplification temperature, typically what happens is that you denature the two DNA strands, primer binds, that is called as annealing step, then you amplify, and then again after the two strands are formed, so after the two strands are formed here, they are again denatured and again they enter the cycle. So it cycles between denaturation, annealing and amplification n number of times, and those many number of times then the strands get amplified. So so you need an instrument for that which is called as PCR, or in case of quantitative PCR it is called as RT-PCR. So the test proceeds, as I said, that the test proceed, you have RNA which is converted to DNA and then it is PCR amplified. So what happens in case of corona? Now it is clear that in the sample you are going to detect presence of viral nucleic acid by extracting the RNA, converting that RNA to DNA, and then amplification of DNA. So what is the sample which you take for this? Typically for coronavirus, you take oropharyngeal or nasal swab. So you take a swab from the deep inside the nose of the infected individual, or you put a swab inside the throat and take a throat swab. That is because virus is supposed to reside there. If there is some virus which is infecting the blood, then one would take blood sample. But in this case, since the virus is known to infect nasal cavity or oropharyngeal cavity, we take oral, throat or nasal swab. That nasal swab, since the test is not carried out where the patient is, even if the patient is hospitalized, the test is not carried out immediately in the same ward. Many times, especially in the case of corona, since the diagnostic tests were carried away in selected laboratories and the samples received from far away places, at times the time for transfer, like from villages and remote areas, the sample time taken between transportation, sample collection and the diagnostic laboratory was few hours, extending up to 24 hours. The virus and the virus RNA should remain intact during that period. So for that reason, the swab is put inside a solution which is called as VTM or viral transport medium, where virus remains stable in that solution, and then it is sealed tightly so as to avoid any leakage, and then it is sent to the diagnostic laboratory. Once received at diagnostic laboratory, this is opened, and then a aliquot of that is taken for the test. From that sample, then one would extract the total RNA from the virus, and there are many methods to do that. Today there are many commercial kits are available, so you don't have to do much. Just use the kit. Solution 1 for 5 minutes, solution 2 for 10 minutes. There are different protocols. So use those kits and prepare the viral RNA. There is a step of checking the quality of the RNA, but in diagnostic test, in many times that step is omitted to save the time. And then you do a first round, convert the RNA into complementary DNA, and that DNA is then amplified in using PCR, and in this case it is quantitative PCR. So as the more and more DNA is amplified, double-stranded DNA is formed, it is quantified, and you see typically you see this curve. So if this sample has no RNA, you will see no virus, no viral RNA, then you will not see any amplification. It's a negative test. But if there is virus, as I told you, one molecule makes 2, 2 make 4, 4 makes 16, 16 makes 64, then there will be an exponential amplification, and in that case the test is positive. Now the height of this curve will depend on, as I said initially, the virus load. So if the virus load is more, the curve will be bigger. If the virus load is smaller, the height of the curve will be shorter. So there is a threshold value. So if you decide that if the curve goes beyond this point, then it is positive. If the curve is below this point, then it is still considered as negative. So this is called as a threshold value. So I'll not go into the technical complications of that unless somebody is interested. So this is how typically a qPCR test for the virus detection is done. So as I told you that this is the structure of the virus, membrane, then there are proteins attached to membranes, spike protein. There's a lot is being talked about spike protein now, because this is the protein which attaches to the human cell and then goes inside the cell. So this is the spike protein. Then there is a membrane protein. Then there is a envelope protein which is present here. And then there is a nucleocapsid protein and the viral RNA. And all the genetic information for making all these proteins is residing inside the viral RNA, which is a single-stranded molecule. And this RNA then codes for all these genes: spike protein, membrane protein, nucleocapsid protein, so on and so forth. So in a diagnostic test, one could choose any one of these proteins and develop a qPCR based diagnostic test for these proteins. Now, which protein you choose, there are many reasons for that. There are some proteins where the sequences change very often. So that protein is not good for the diagnostic test. And so one has to do a lot of exercise to decide which protein and which region of the protein is going to give you a accurate diagnostic test. The selected protein should also be specific for the virus and should not show any cross reactivity with the related virus. Because as you know, the symptoms from corona are common. It's like cough, cold, fever. So any other virus which causes similar symptoms, it should not come up positive. The test should not come positive in such infection. It should come positive only when there is a specific infection from coronavirus. So typically most tests use this ORF1 region as the target for developing diagnostic tests. And then they also use ribosomal dependent RNA polymerase, RDRP gene also is used. So in many tests, the test detects the presence of, instead of just one, to make it more specific, it detects the presence of both the genes. So both the genes are quantified in a single test, and the test is considered positive only if both the genes are turned out to be positive. So, since the corona pandemic hit, a lot of exercise has been done in developing diagnostic tests, and in India, Indian Council for Medical Research is the regulatory body which authorizes the diagnostic tests. So ICMR has a mechanism by which, if you develop a new diagnostic test, it has a mechanism by which it tests your test against the other their standard test. It determines its specificity and sensitivity, and only if the test passes those specificity and sensitivity criteria, that test is approved as a diagnostic test. So as of today, these are the, this information is for reference date for this is July 2020. So as of July 2020, the diagnostic kits made by these 66 suppliers was approved by ICMR for the diagnostic RT-PCR test in India. So test kit produced by any of these 66 commercial suppliers can be used by a laboratory for doing RT-PCR based diagnostic test for coronavirus infection. So the test, the kits vary, as I said, in terms of the specific region and the primers which they use for detection of coronavirus, and other methodologies like enzymes they use. Some laboratories have used some different enzyme where the amplification could be faster and you get the results quicker. But typical time for the detection of infection using RT-PCR is 3 to 4 hours. So it takes some time for the extraction of RNA, and then there is a PCR amplification. So typical time taken is 3 to 4 hours. People have also tried to develop tests like if you have read the newspaper, you have read about dry swab. So wherein you don't have to transport the swab in viral transport medium, and you don't have to do the RNA extraction. That swab can straight away be used for RNA PCR. So in India, CCMB has developed that technology, and which has now been approved by ICMR. So then that time taken for RNA extraction is saved, and transportation of swab from the testing site to the laboratory becomes easier because it's a dry swab. You don't need viral transport medium for that. So the tests vary in, the typical time taken is 3 to 4 hours to get the results of a diagnostic test. I talked about different primers that are used. So just to give you some idea, I said that these are different manufacturers use different genes. Here you can see this Altona uses spike protein and envelope. This uses ORF1. This uses RDRP and spike protein. I also said that many diagnostic tests use two genes. So this is RDRP and this is just RDRP. This is only envelope. This uses 3 RDRP nucleocapsid and envelope protein. So like that, many different manufacturers have different genes as targets, and further for a particular gene, different region of the gene using different primers is amplified. Another technique that has been being used of late in the diagnosis is the genome sequencing. And the genome sequencing has many advantages. It helps to identify the conclusive identification of causative agent. But not only that, if you know the entire genome sequence of the virus, it helps to understand how the virus is being spread in the community, whether it is getting transmitted only within the community or there is some external source. And then it also lets us know how the virus is changing. So that helps in development of newer diagnostic tests, or it has implications on design of vaccine and efficacy of vaccine. So, and why genome sequence is feasible today is because modern day the genome sequencing technologies have become very fast and very cheap. So large scale genome sequencing is possible. So genome sequencing, as if you read the newspapers, genome sequencing has helped us a lot in understanding the spread of coronavirus in our country. So the first definitive diagnosis of corona infection also came from genome sequencing. So in March, after the infection was reported in China in December, the genome sequence of the virus was determined immediately. As you can see here, it was submitted in the first week of January. So, but the analysis was done much before that. So immediately in January, within a month, the entire genome sequence of the virus was known, and based on that sequence only, they analyzed the sequence and compared that with other known virus sequences, and based on that only, it was concluded that the causative agent for the respiratory disease that people from Wuhan were suffering, the causative agent for that is a coronavirus, and then it was named as SARS coronavirus 2. So the first definitive diagnosis of corona infection indeed came from genome sequencing, and it also helped us to conclusively understand the bad origin of virus. Here again, you will see from 5-6 more genome sequences, you can see that the patient viral genome sequence matched very well with bad coronaviruses. So this also helped to conclusively pinpoint the origin of infection. So that is the advantage of genome sequencing technology. And as of today, you will see how the virus is evolving. So this is a global data that is available. And today, more than 1.5 lakh viral genome sequences are available globally. And then it tells you how the virus is evolving. So the red dots are, red and yellow are some of the early variants of the virus, and blue ones are the present variants of the virus that are in circulation. And you can see that currently these are most of the variants that are in circulation today. Looking at the India, initially the study was done 3 months back. It was shown that different parts of India, the predominant variants of the virus were different. So a different picture was seen in western part than in southern part than in northern part. So different variants were prevalent in different parts of the India. But over the time, the picture started to change, and just a quick glance, you will see that whether April or May, only one type was predominant in India, that is 19A was predominant in Delhi. Whereas if you take example of Telangana, in April, it was 19A type that was prevalent, but the picture changed in May. You do not see any more 19A, but you see more of 20A and 20B. If you look at West Bengal, in the month of April, 20A was predominant with little of 19A, 19B, and in May then 20A was predominant. So it helps us also to understand how the virus genotypic changing. And if you look at our results from Maharashtra, again, these are the samples from April, May, and this is June, July. And you will see that in all the 3 districts we studied, the picture changed from what it was in April, May to what you see in June, July. The other, for example, in our own Pune district, other 2 variants, 19A, 20A, are not there at all. Earlier, they were present as a small percentage in April and May, but June, July, you see completely taken over by 20B. Similar is in this district, 19A has gone completely, taken over by 20A and 20B. And here again, you see very little of 20A, majority being 20B. So thus you can find out how in the population, the type of the virus that is circulated is changing using genome sequencing. Now, recently, the antigen test has been developed for the detection of coronavirus. And this test, as I said earlier, it is based on the detection of virus-specific protein. So you don't look for the viral RNA, but you look for a virus protein in your test sample, which is again a nasal swab. So again here, the protein should be specific for the virus, and it should detect only specific coronavirus virus and not any other virus. So this is the antigen is basically a protein, and the test is based on specific detection of coronavirus specific protein in the patient sample. So how the test works is that there is a specific protein and against which there is antibody. The antibody binds to the protein, and then this bound antibody is then detected by fluorescence or immunology or colorimetric reaction. This second antibody detects the first antibody. So there is an antigen to which the antibody is bound. So this is an antigen to which antibody is bound, and then this specific antibody is then detected by another antibody to which the detection mechanism is attached. So here again, typically you take the swab, put it inside your sample tube, and then this is a test which actually can be carried out on spot. So you don't have to take the sample to a specialized laboratory. On the spot, there is a strip on which you put the reagents, and then there is a control in that, and you incubate that, and the color develops. So presence of a specific color band indicates the presence of virus. The band doesn't develop, it's an absence of virus. So basically it has, the strip has coated the antibody against the antigen, then the control antibody, and then antibody to which anti-antibody to which is linked to a detector molecule. So when you spot the sample starts moving by lateral flow, and then if there is a specific antigen antibody reaction, the binding will take place here. Here, of course, the control binding will take place, and then as it moves further, you add the reagent and develop the strip. Now, if the specific antigen is present here, you will get a band here. And this serves as a control because to indicate that everything else with the reagents was good and the reagents are not incorrect, this band develops. So whether the test is positive, if the test is negative, only this band will develop, and if the test is positive, 2 bands will appear. So appearance of these 2 bands is indicative of positive test. Appearance of only 1 band indicates that the test is negative, but your reagents are correct. If this band also does not appear and the strip is blank, then you cannot rely on this test because there is some problem with your reagents. So if you get 2 bands, test is positive. If you get 1 band, the test is negative. So this is a colorimetric method. This secondary detective antibody is the place where the detector molecule is attached and you detect this molecule by using a colorimetric reaction. So that's how the test is based. The advantage of this test is this is rapid. It can be done in 15 minutes. It could be done on the site. So you take the sample from the patient and immediately carry out the test there. So that is the advantage of this test. And currently ICMR, as of August 2020, these 3 tests are approved by ICMR for rapid antigen test. These 3 test kits manufactured by these 3 manufacturers are approved by ICMR for rapid antigen detection test. However, there are some issues about sensitivity of the rapid antigen test. So what is done is, if the rapid antigen test is positive, then one can be sure of presence of infection. So it is positive means the person definitely has infection. However, if the test is negative, but the person is still suspected of infection, then RT-PCR is recommended, because RT-PCR is still considered as a gold standard for the risk of coronavirus infection. So rapid antigen test for screening, if it comes positive, yes, the person is definitely infected. If the test is negative, but the person has classical symptoms of coronavirus infection, or the person is definitely in contact with corona infected persons, then RT-PCR test is still recommended, which is a gold standard. So the third test is looking for antibody. I'm not going to talk much about that. It is primarily used for presence of antibody in the patient serum, and it is presence of antibody in the patient serum is indicative of a person having infection, person having a past infection. And this test helps to detect those who had asymptomatic infection. So there are many cases where the person has infection but does not develop the symptoms. So person doesn't know that he had infection. So, but for the surveillance purpose, it is important to know who all had the infection. So this test is done for serosurveillance, and it is not the diagnostic test for coronavirus infection. So I will not talk much about that. And with that, then I'll end my presentation. Thank you very much. Thanks for giving me this opportunity. And I will be very happy if there is time to answer a few questions.
Thank you very much, sir. It was a very clear presentation. We all enjoyed, and I'm sure the students also must have understood it very well. Thank you.
I request Dr. Mahendra, Biotechnology Department, to give quick response to Sir's lecture. Dr. Mahendra, over to you. Dr. Mahendra.
Hello.
Yes.
Hello.
Hello.
Good afternoon.
Yes.
Good afternoon, sir. Thank you for giving this opportunity. I'm very happy to see the lecture given by sir. It is very impressive. Sir, from my hand, being a biotechnologist, I want to ask you a question that why PCR, why RT-PCR is not being widely employed in detecting the, positive cases with regard to corona. Most of them, they are using rapid antigen kits. However, the results may not be that much sensitive to my knowledge, and I feel that it is to better go for RT-PCR, tests. Why that thing is not being continued in? That is not in practice.
So the answer to that question is, see, as I said repeatedly, RT-PCR remains gold standard for the diagnosis of corona.
Sir.
The rapid antigen test has the advantage that it is cheaper and it is done quickly. So the situations where a large number of quick diagnostic tests need to be carried out, in those situations, the rapid test has the advantage that it can be done in 15 minutes and at much less cost. So that is the reason for large scale screening, rapid antigen test is done. But for confirmatory diagnosis, RT-PCR is the accepted test. So it's the gold standard. Yeah.
Sir, thank you, sir. Thank you very much.
Thank you.
It's a time for next lecture by Dr. Vinita ma'am.
Thank you very much.
I request Dr. Jalandhar to introduce resource person. Dr. Jalandhar,
Okay,
over
sir.
to you.
Okay, sir. Thank you, sir. Very good afternoon to all. It's my pleasure, immense pleasure to introduce Professor Vinita V. Deshmukh ma'am. She is working as Head Department of Sanskrit Samhita Siddhanta, BSDTS, Ayurved Mahavidyalaya, Wagholi, Pune. And also she is working as the Deputy Director of BSDTS Integrated Cancer Treatment and Research Center, Wagholi, Pune. She did her BA-MS degree in 1994 from Mumbai University. She has completed her Doctor of Medicine in 1998 from same university. She did her PhD degree in 2006 on Kaya Chikitsa from Pune University. She has completed PGDCR degree in 2006 on clinical research from ICRI. She has completed diploma course in Ayurvedic dietetics from Tilak Maharashtra Vidyapeeth, Pune. She has total 21 years of teaching experience. She served as in charge Ayurvedic courses of BS DTS Ayurveda College since 2005 to 2012, and also Professor Vinita ma'am invited to conduct Ayurvedic courses at Ayurveda Academy of Yoga in daily life in Vienna from 2005 to 2012. And she worked as a research fellow for cancer research project from 1996 to 2005. And also she worked as in charge for cancer research project 2006 to 2010. Then Professor Deshmukh ma'am worked as in AIDS research projects ICRTC from 2099 to till date, and she has presented various research papers in state level, national level, and also international level conferences on the topic Ayurveda and cancer. She has organized several camps, exhibitions, lectures, awareness programs on cancer and Ayurveda at ICTRC. She also organized seminars, workshops related to cancer and Ayurveda. Total 4 students completed their PhD degree under the guidance of ma'am, and one student completed MPhil degree under the guidance of Deshmukh ma'am, and total 9 students completed their MD degree, and 3 students are working for their MD degree under the guidance of Deshmukh ma'am. And also she has the co-author of some books. Number 1 is Cancer Ayurveda and Cancer Sati Lok Prayogi Ayurveda. This is, I think, Marathi. A handbook on cancer, Better Tomorrow for Cancer with Ayurveda. These are the 4 books wrote by ma'am, and also she wrote many articles in newspapers, magazines, periodics on cancer Ayurveda. She is expertise in clinical research in the field of cancer and Ayurveda, clinical research in the field of HIV, AIDS, and Ayurveda, and literary research. She has felicitated by different awards like Jivak Award 1993 and Vaidya MB Pulatkar Research Award, that is in 2008. Thank you very much, sir, for giving me this chance, this opportunity. Over to ma'am.
Vinita ma'am.
Hello.
Yes, ma'am. You can start your screen sharing.
Yeah. Hello.
Yes, ma'am. You're audible.
Yeah. Can you see the screen?
Yes, we can see.
Hello. Can I start now?
Yeah, you can start, ma'am. Now it is visible.
Yeah, now it's visible. Okay.
Yeah.
I am very much grateful to Telangana University Department of Botany for giving me the opportunity of sharing my experiences about COVID-19 and cancer patients. So, my today's topic is COVID-19 Ayurvedic perspective and experience of cancer patients under long-term Ayurvedic treatment in COVID-19 pandemic. We have our integrated cancer treatment and research center at Wagholi, Pune. The center is a center of excellence of Department of Ayush and supported by Department of Atomic Energy for Radiotherapy Department and affiliated to Indian Cancer Society. It's also supported for research activities and Panchakarma Hospital by Tata Trust. Param Pujya Prabhakar Keshav Sir Deshmukh Maharaj was a person who started this project at Wagholi, and he was the authority in Indian medical science, that is Ayurveda. His son, Dr. S.P. Sir Deshmukh, he is also an Ayurvedic physician, well-known Ayurvedic physician. He is first PhD in Maharashtra in the subject of Ayurveda. He and Dr. Arvind Kulkarni, who is a radiation oncologist, ex-radiation oncologist of and the HOD of radiation oncology department in Bombay Hospital, both of them started cancer research project at Wagholi, in which cancer patients are treated with adjunct Ayurvedic treatment. They are taking chemotherapy, radiotherapy, and surgery, and along with that, they are taking Ayurvedic treatment. This is our team of doctors who from all our centers from Pune, Mumbai, Kolhapur, Solapur, and Nashik. So, as we all aware of the fact that cancer is a leading cause of death in the world, in 2018, 18.1 million new cases, 9.6 million deaths from cancer, and the conventional therapy for cancer is surgery, chemotherapy, and radiation. But it has several side effects like acute, intermediate, and late. Due to disease itself and due to conventional treatment, cancer patients have weak immune system, and due to which they are prone to develop various infections, including COVID-19. So they are at high risk like diabetic and hypertensive patients. This is statistics of COVID. I will not go in detail because all of us are aware of these statistics. Now, as we have seen, older people and those with underlying medical problems like cardiovascular disease, diabetes, chronic respiratory disease, cancer, and those have habit of addiction like smoking, alcohol, drinking, are most likely to develop serious disease, including COVID. There are various publications from various well-known journals regarding association of COVID-19 and cancer. So this is one publication which I have quoted here, but there are many publications regarding this topic. We are all aware of the fact that this disease mainly transmits through droplets of saliva or discharge from nose. As a preventive measures, there are various preventive measures like washing hands, maintaining at least 1 meter distance, and everything that we all aware of that fact. But as of now, in modern medical science, exact treatment for COVID-19 is not available. Symptomatic treatment is recommended as similar to other viral infections. Now, what we have seen that what we have to see today is what is the similarity of COVID-19 and the diseases mentioned in Ayurvedic text. As we all aware of the fact that Charaka Samhita and Sushrut Samhita are the oldest Ayurvedic text, which has mentioned various diseases which we are facing right now. And only the thing is that the names of these diseases are different, but the symptoms are same. So in Charaka Samhita, a concept of contagious disease or communicable disease is elaborately mentioned. And similar concept is also mentioned in Sushrut Samhita, but it is labeled as Aupasargik Roga. And it is given clearly in Sushrut Samhita that skin diseases, fever, tuberculosis, and conjunctivitis, these are the four major diseases which are the contagious diseases, highly contagious diseases, which spread from one person to another person. And their mode of transmission is also explained in Sushrut Samhita as sexual actions, touch, respiration, eating together, sleeping close to each other, using clothes, garments, and cosmetics of each other. So, as of today, if we see the major symptom of COVID-19 is jwara, that is fever, and its mode of transmission is explained as through respiration, through touch, using clothes, garments, which are seen nowadays. Similarly, in Ayurvedic texts, in Charaka Samhita, there is a description of epidemics under the term of Janapadodvamsa, where it is mentioned that due to either of deranged or contaminated air, water, habitat, and season, certain diseases arise which kill mass of people. Further, it is mentioned that there are underlying causes for epidemic diseases like not following ethical code of conduct by majority of people in the society and even some external factors like Krumi. So this concept of Krumi is well elaborately mentioned in Ayurvedic Samhitas, which can be correlated with the concept of bacteria and vector and viruses nowadays. The treatment recommended in Ayurvedic text for Aupasargika Roga and diseases originated due to Janapadodvamsa are mainly two types of treatment, that is Rasayana Chikitsa. That means the medicines which enhance immune system, and secondly Panchakarma, which detoxifies the body. Ayurvedic Samhitas elaborately mention concept of Rasayana, that is immunomodulators, and use of specific Rasayana medicines in specific diseases. There is a list of Rasayana medicines, a range of Rasayana medicines mentioned in Ayurvedic text, but the physician should know, the Ayurvedic physician should know the properties of each Rasayana medicine, and accordingly he has to treat the disease, specifically the Aupasargika Roga, that is communicable diseases. Now, BSDT, that is our institute, has an experience of treating non-curable, dreadful diseases, especially the diseases which have hampered immune system, and the medicines are used for boosting immune system. So along with cancer, we have a HIV/AIDS project. So we documented efficacy of adjunct Ayurvedic treatment in HIV/AIDS and cancer patient in improving their quality of life, boosting immunity, and increasing survival. So Ayurvedic formulations used in HIV/AIDS and cancer patients are found to be enhancing their immune system. It is seen that if HIV/AIDS patients and cancer patients are treated with Rasayana Chikitsa, which is mentioned in Ayurvedic texts, for long duration, definitely in HIV/AIDS patients, their opportunistic infections are remarkably reduced. Their CD4, CD8 counts are either maintained or increased, and in both patients, cancer and AIDS patient, the quality of life is remarkably improved. These immunomodulatory properties and reducing oxidative stress properties of these medicines are tested in our laboratories, our immunology laboratory and our molecular biology laboratory, with certain parameters like cytokines and ROS parameters. Many times there is a question, because these Rasayana medicines are not only herbal medicines. These medicines are herbal, mineral, as well as metallic medicines. For example, all of you are aware of Shatavari. The Shatavari is a herbal Rasayana medicines, or aloe vera, or guduchi, that is Tinospora cordifolia. So these are the herbal medicines which have a Rasayana, that is immunomodulatory properties. Similarly, there are some mineral medicines like Mauktik Bhasma or Praval Bhasma, which has immunomodulatory property. There are some Ayurvedic medicines in the form of metals, but prepared in a specific fashion to detoxify these metals and to enhance their properties. So some medicines in the form of metallic medicines also possess immunomodulatory properties, which include Suvarna Bhasma, Raupya Bhasma. So these medicines are very useful in the treatment of immunomodulation. But when we use these medicines in the form of minerals and metals, many times there is a question raised by scientific the people in other fraternity, that whether these medicines are safe or not. So for that purpose, in our hospital, we have a drug standardization laboratory funded by Tata Trust, in which laboratory we standardize these medicines on modern yardsticks, uh, in, like for example on ICP, or in the form of HPTLC or HPLC. So the tests which are not done in our laboratory are outsourced in accredited laboratories like CSIR, NCL, IDRL, and ACTRAC. The medicines which are specially metallic medicines, for these medicines we are also doing the animal toxicity studies at ACTRAC. So this is a background, or this is just a background how we think from Ayurvedic point of view for any disease. Now, what we have done, that the COVID-19 started from March 2020. So in the month of November, at the end of November, we have done an extensive survey of our cancer patients, that how many patients of how many our patients have developed COVID-19. Now, before giving brief information about this survey, I want to just share one slide with you, that this is a pattern of Ayurvedic treatment at our center. All cancer patients confirmed with biopsy are coming to our center and divided in 4 groups. Group A is a group of patients who do not wish to offer any conventional treatment like surgery, chemotherapy, and radiotherapy. This is a small group because we never insist patient to undergo only Ayurvedic treatment. Ours is an integrated center, so we convince them to take conventional treatment along with Ayurvedic treatment. But due to old age or maybe due to financial problems, sometimes patients are not ready to offer any conventional treatment. So in such patients, we treat this patient, we call this type of treatment as Vyadhi Pratyanik Ayurvedic treatment. That is the Ayurvedic treatment which directly acts on Vrana Granthi or Buddha, the similar diseases to cancer mentioned in Ayurvedic text. The second group of patient is a group B, in which the patients are already treated with conventional treatment and now they have a recurrence. So this is a group of patients who are having a lack of immune system. So in this group, Rasayana Chikitsa plays an important role. Then C group is a group of patients who are taking conventional treatment and Ayurvedic together uh, hand to hand. Along with conventional treatment, they are starting Ayurvedic treatment. So our main focus of treatment is to minimize side effects of chemotherapy and radiotherapy and to boost up immune system. And the last group, that is D group, is a group of patients in which patients are previously treated with conventional treatment. Now they are in remission and they opt for Ayurvedic treatment just to prevent recurrence. So in this group, we administer Shamana Chikitsa, Rasayana Chikitsa, and Panchakarma, all three types of modalities of Ayurvedic treatment to improve their quality of life and to increase disease-free survival. So these are the categories of patients coming to our center, and these categories are done in view of or giving focus on Ayurvedic treatment. So if we'll go for the survey, that if we'll see that these D group patients and C group patients are more in our center, and they are treated for a long duration. So Ayurvedic treatment is not like a chemotherapy or radiotherapy, which is a time-bound treatment. This treatment basically improves quality of life of patient and increases immune system. So we have to administer this treatment for long duration, minimum for the period of 3 years to lifelong. So this is the basis of our treatment. So the survey was carried out in our center, in which the patients from all these four groups, A, B, C, D, were taken into consideration, and the criteria was they must have taken at least 1 year of Ayurvedic treatment, because before that we cannot assess their the response of the Ayurvedic treatment. So the survey study of total 649 cancer patients was done to assess their health issues during this pandemic. These patients were earlier guided for healthy diet and lifestyle modification from Ayurvedic perspective. And the data was analyzed on the basis of sex-wise distribution, age-wise distribution, type of cancer, history of diagnosis of COVID-19 in their near contacts. So this survey was carried out just to see that is what is the impact of Ayurvedic medicine on these patients. Among these 649 patients, this is a picture which we can see that we have our patients from Pune Center, Mumbai Center, Kolhapur Center, and Nasik Center. So altogether, there were 649 patients. And if we see the patients who develop COVID or who develop corona disease or who were COVID positive were only 27. So this percentage is 4.16%. And among these 27 patients, 3 patients expired. That is, the death toll was just 0.46%. Now, in next slide, I will show you the distribution of these patients. So among these 27 patients who had COVID positive history, among them 13 were male, 14 were female. Similarly, breast cancer patients were more. 11 patients were had suffering from breast cancer, 4 from oral cavity cancer patients, 2 of leukemia, 2 of NHL, 2 of urinary bladder, and 1 each from prostate, ependymoma, GIST, colon, esophagus, and metastasis in lymph node. If we saw the distribution of age, the minimum age was 3 years. A girl having leukemia, 3 years old, died. Sorry, she had she was corona positive, and maximum age was 73 years. We distributed these 27 patients according to symptoms. Among these 27 patients, 6 were asymptomatic, 15 had mild symptoms, 3 had moderate symptoms, and 3 had severe symptoms who expired in the hospital. Apart from this study, what we have studied that among these 649 patients, how many patients had signs and symptoms of upper respiratory tract during this pandemic? Because it's the possibility that maybe some patients have not done their RT-PCR test. So what we found that 35 patients had upper respiratory tract symptoms apart from these 27 symptoms. But the notable finding was that their symptoms subsided with home remedies. Because before starting or at the time of starting of pandemic, we send diet recommendations, do's and don'ts to all of our patients, and they followed it, and with that they recovered in the symptoms. Now this is just to give an Ayurvedic perspective of cancer. In Ayurveda, cancer is not mentioned as a single disease. It's mentioned as a group of disease like Dushta Vrana, Dushta Granthi, Dushta Arbuda, Dushta Nadi Vrana. And leukemia and Hodgkin's, non-Hodgkin's and Hodgkin disease is similar to that of Jwara, that is Sannipatik Jwar, Rakta Pitta, Pandu and Krumi. As we have seen this slide earlier, I'll go next. Now, these observations that this much less number of patients, cancer patients, had corona positive, were corona positive. So we looked at their symptom, we looked at their treatment, line of treatment. So this is a general line of treatment in our center. We treat all patients with Shamana Chikitsa, that is oral Ayurvedic medicines which bring doshas and dhatus in equilibrium. Second is Panchakarma treatment, that is detoxified treatment. Third is Shashti Upakrama or allied procedures, that is local treatment of ulcers. Fourth and important is Rasayana Chikitsa. For long duration, we treat these patients with Rasayana Chikitsa. I will emphasize on this treatment afterwards because this is a main crunch of increasing immune system. Then we advise them specific nutritious diet, but which is healthy and light for digestion, healthy lifestyle. Then counseling is also a very important part of our treatment and avoiding risk factors. So all these things together are given to these patients. Now here I have to explain that Panchakarma Chikitsa, that is detoxifying treatment, is not given to all of these patients because this Panchakarma treatment needs eligibility. So the patients who are eligible for Panchakarma are treated with Panchakarma Chikitsa. So these are the common treatment, these were the general treatment modalities. This is a specific treatment modality. Now, Panchakarma treatment is 5 types of treatment: Vamana, Virechana, Basti, Nasya and Raktamokshana. Here I mentioned that in which type of cancer, which treatment is mainly administered. So in throat cancer, lung cancer, melanomas, breast cancer, Hodgkin disease, Vamana is recommended. Virechana is recommended in duodenum, pancreas, liver cancer. Basti, that is medicated enema, is given in rectal cancer, cervical, uterine cancer, ovarian cancer, bone metastasis and testes. Nasya, that is nasal administration, is given in nasopharyngeal cancer, brain. And Raktamokshana, that is bloodletting, application of Jalauka or leeches, is mainly given in liver cancer, that is HCC and leukemia. Now, these are the common herbs which we are giving to these patients because see, Ayurvedic treatment is not like a conventional treatment that for breast cancer, this much is a CAF protocol or like or 4-AC plus Taxol protocol. It's not confirmed from the point of view of disease. Mainly Ayurvedic treatment, which was given to all these 649 patients in our project, was a personalized tailor-made treatment. But there is a set of medicines. There are some herbal medicines, mineral medicine, metallic medicines, which we are commonly using to these patients. And what we thought that due to long-term administration of this Shamana Chikitsa, Rasayana Chikitsa, Panchakarma treatment in some patients, these patients definitely have a immunomodulatory action, which not only help them to face the disease like cancer, but to face the opportunistic infections, to face the diseases which are contagious, and this due to this the death toll of these patients was very low in our project. So the herbs, there are various herbs which we use as single herbs, mixture of herbs, Gugul Kalpa or sugar-based Kalpa, Asavarishta or medicated ghee. These are the common medicines which have a pacifying effect on doshas. Now, this is a list of medicine, that is Amalki, Avla, what Indian gooseberry we know, then Ananta, that is Hemidesmus indicus, Kokshur, Guduchi, that is Tinospora cordifolia, Haridra, that is turmeric, aloe vera, and Kamkor. And especially in these type of cancers, we are administrating these medicines. Then Kamal, that is lotus, Lodra, Pushkarmo, Parivadra, Shatavari. This is almond, Vasa, that is Adatoda Vasika, and Yashtimadu, licorice. Then in mixture, there are Triphala Churna, Sitopaladi Churna, Talisadi Churna, Invastak Churna, Navaya Slova, Praval Pranchabhut, and Kamrudaras. They are given in specific types of cancers. Then there are some herbo-mineral metallic medicines like Arogyavardhini, Chandraprabha, Shankhavati, Vasadivati. This Arogyavardhini is commonly used in pancreatic cancer, liver cancer, and leukemia. Chandraprabhavati is mainly given in breast cancer, uterine cancer, ovarian cancer, bladder cancer, kidney cancer. Vasadivati is given in oropharyngeal cancers. And there are certain Guggul Kalpa, that is, Guggul is Comifera mucul. And Sushrutaracharya specially mentioned that Comifera mucul has a rasayana property, and along with that, it has a property of reducing wounds or ulcers or arbuda. So many patients from our these 649 patients, they are treated with various types of Guggula for which they are indicated. Then Kvathas, that is Kumari Asau, Varnadikvat. Then these are the Kalpa, that is sugar-based medicines like prepared from Shatavari, Hemidesmus indicus, Adathoda Vasika, and almond. There are various types of ghees, medicated ghee. As we know that medicated ghee, cow's ghee, has a best property of rasayana. So in Ayurveda, there is a tradition of mixing these gruta or cooking them with various herbs. So when we cook them with various herbs, their properties are enhanced. So there are Yashtimadu gruta, Padmakadi gruta, Vasa gruta, which are given in various indications of cancer. Also for local application, there are some oils like Nirgundi tel, Karanza tel, Yashtimadu tel, Paribhadra tel. Then comes very important thing is rasayana. So most of these patients are treated with Suvarna Kalpa. Suvarna, that is gold, is a treatment of choice which has a rasayana action. So in most of our patients who need rasayana or immunomodulatory treatment, we give them various forms of gold. So we treat them with Raupe Suvarna Suchekar or Suvarna Suchekar or Suvarna Malini Vasanta, Yakrut Yodarari Loha. There are n number of medicines in Ayurveda which has which possess immunomodulatory property due to addition of Suvarna Kalpa, Suvarna Bhasma in these medicines. And I must mention that in our drug standardization laboratory and in the laboratory of NCL, we tested Suvarna Bhasma, which is prepared in our own pharmacy. It takes at least 2 to 2.5 months to prepare Suvarna Bhasma, good quality of Suvarna Bhasma. So we are preparing it in our own pharmacy, and we not only preparing in our own pharmacy, we are giving it for drug standardization in labs. And what we have found that the Suvarna Bhasma, which is prepared in our pharmacy, has come up to the level of nanoparticles, and not only that, it is safe and effective. So all preparations of Suvarna Bhasma are very important in management of cancer patients, and it is important to prevent the disease. And what we think that the same Suvarna Kalpa we are also giving in HIV/AIDS patients. So they have a very great immunomodulatory action. And what we think that very less toll of death in our patient is mainly due to these Rasayana medicines. So these Rasayana medicines which I have mentioned earlier are the Suvarna Bhasma, Raupya Bhasma, Almond, Triphala, Yashtimadu, Pippali, Piper Longum, Abrak Bhasma, Shatavari, Gokshur, and Kumari. So these Rasayana treatment is very effective for immunomodulation, and I think due to administration of these type of medicines, most of our patients remain disease-free or have developed mild symptoms. So this is the conclusion. In view of high risk of developing COVID-19 in cancer patient, minimum number of cancer patients in our study were diagnosed with COVID-19. I already quoted the number, only 27 patients. Most of them who were diagnosed with COVID-19 were either asymptomatic or had minimum symptoms. Death toll was very also insignificant. Only 3 patients died with COVID-19. So this survey study primarily demonstrates Rasayana action or immunomodulatory effect of Ayurvedic treatment in cancer patient, which not only helps them to fight against cancer, but to prevent from other communicable diseases. Thank you.
It was a wonderful presentation, and now we know how rich is our Ayurveda medicines. It's really very nicely explained by you, and especially in cancer and COVID patients, how we can tackle it. I have one question, which is not directly connected, but to increase immunity, are there some Ayurvedic medicines can be used for this COVID to prevent? For preventing, are there some drugs or medicines which can be used for preventing COVID? Increase immunity.
Yes. See, what we have seen that COVID-19, this virus mainly attacks respiratory system. So in Ayurveda, there are specific Rasayana medicines which boost the activity of respiratory system. The main of them are Pimpali, that is Piper longum, then Yashti Madhu, that is licorice, and Abrak Basma and Suvarna Basma. These four are at priority for preventive COVID.
One question I have. Piper longum, we have used it for anti-fertility studies, I remember, long back. So whether it has got some anti-fertility effect also. Actually, it was with Emilia ribus, we had used it. Piper longum and Emilia ribus. This was actually, I was in ICMR, and ICMR had suggested this to be used to study the anti-fertility effect. So whether it has got anti-fertility effect also. So that is what maybe then it may not be.
Yes. Many Ayurvedic medicines, they have multiple uses. Actions, so Pippali, Piper longum has also have anti fertility effect. But the important thing is that how you use it, with which medicine you are using, the mode of action depends on that.
Good afternoon, ma'am.
Yes.
It was very sound to listen the great Indian field of medicine, which has been being forgotten by the young generation and youth. It was very wonderful to listen the entire experiences of cancer patients during the pandemic and the patients. It was very surprising to see the numbers. Magical numbers of patients were found to be the affected cases, and among them survival rate was very high. So it is a lesson we learned today. I think among the participant group also we have understood very clearly. So in our school studies, we got a stanza in a poem that Pogadara ni talli bhumi bharati. In that line, poet says that praise the motherland India. So by seeing too many things, we need a focus, a care to observe our surroundings and what India is actually missing in the field of modern lifestyle. So it is a great number of patients are being treated in the center at different area in different locations, and it was very important to note among all the students and participants of this workshop that Ayurveda has got great potential, and from the experiences of ma'am, live experiences of ma'am during the pandemic, we have learned a great thing that Ayurveda is long life or long live Ayurveda forever, and the Indian medical system need a lot of appraisal without any injectables and without making the patients in days of pandemic. The allopathic medicine or allopathic hospitals are charging lakhs of rupees once you're admitted in the hospital. Whatever be the problem, health ailment, whatever be the health ailment, the major thing what we noticed during pandemic was people were charged lakhs of rupees when they got admitted to the hospital, and the healthcare workers are also it was a bad time for general public, and if people start towards our indigenous system, what we have great thing. So with this lecture we have learned a lot, and I feel our students who are participating and the participants who are from nationwide will also have got a clear-cut ideology that Ayurveda is the great boon, where the patients, whatever will be the pandemic and whatever will be the situation, it will flourish for a long time, and we will get a lot more information inputs by ma'am in future also. Whenever we hold any meeting offline or online, we expect that Deshmukh ma'am will be with us, and she will be encouraging and making the people, audiences with her loud experiences, what she shared today. Really, it is a note of happiness that a great job has been rendered by ma'am, and the great information on Ayurveda, and a clear-cut, eye-opening presentation was witnessed today. And I thank especially the team of the Trust Hospital, and ma'am, and Tarla ma'am also. Tarla ma'am is also equally thankful. Wholeheartedly we express thanks, as she was the person who roped you as a resource person, and we feel it is a good opportunity to listen you at this moment. And I feel that our participants will also knock towards the Ayurveda and get the benefits of Ayurveda in the life. And also we can fight with Corona. India is, that's why the death toll or fatality in India with Corona is less, though there is a more infection rate. Though the infection rate was very high, still the fatality or mortality, morbidity was less, because of such systems are still existing in our in and around the environment. So with these few words, I thank once again the convener ma'am and the resource person of today, Dr. Vinita V. Deshmukh ma'am, and I request Aruna ma'am to give her remarks on the presentation. Aruna ma'am, over to you.
Recording आहे ना?
Aruna ma'am. Aruna ma'am. Vidya ma'am.
Good afternoon, everyone. Thank you, Halim Khan. And good afternoon to the today's distinguished speakers and the participants. It was a very nice afternoon session where Dr. Yogesh sir and Dr. Vinita ma'am has given us so many insights regarding COVID. And the ma'am has...
Ma'am, your camera, you switch on your camera. Video is off. Yeah.
And Vinita ma'am also has presented very well. And ma'am, we really appreciate knowing some things about Ayurveda which we didn't know, because indigenous medicine is now being more popularized, and we are very glad about it. And we are very glad to know that there are so many institutes in India where we are using all these indigenous medicines. Ma'am, it was really an awesome lecture. Even sir lecture also was good. It was given a lot of insights to all of us. And I thank the academies for the lecture workshop, as well as I thank the convener, and I thank Dr. Halim Khan. Thank you.
Aruna ma'am, now you.
Hello.
Yes,
Hello.
you're audible, ma'am.
Yes. Just in
Aruna
last,
ma'am.
yes, in last sentence, just I want to thank all of our cancer patients, because they have a huge faith on Ayurveda. So they followed all the Ayurvedic principle during this pandemic. And I encourage all the Indian patients of Ayurveda, of cancer, not only Indian but patients from abroad, to follow the Ayurvedic principles and follow the Ayurvedic lifestyle to get healthy life. So I thank all of our patients and my team of doctors and non-medical person who work throughout the period of COVID-19 in this pandemic for these patients. And I also thankful to Dr. Arvind Kulkarni and Dr. Sardeshmukh sir. I also thank to Nishigandha Naik madam and Dr. Vidya Gupta madam who gave us boost up for validating Ayurveda on modern principles. Thank you.
Aruna ma'am, over to you.
Madam, unmute.
Ma'am, you unmute yourself, ma'am.
Unmute, madam. Button.
This mic is there. You pick you. మ్యామ్ మైక్ లా ఉంది కదా, అది ఒకసారి ప్రెస్ చేయండి. ఒక క్రాస్ లైన్ ఉంది మైక్ పైన. ఇక్కడ మైక్ లాగా ఉంది, మ్యూట్, అది
డబుల్ క్లిక్
ప్రెస్ చేయండి.
I'm not able to unmute. You should self unmute.
Yeah, I have.
Haleem Khan sir, you try, ma'am.
No, from our side, I can ask to unmute. She has to do unmute.
Yeah, I also tried, but
In this only we can. She should. Ma'am should do it actually.
yeah.
Now you speak, ma'am. Your Aruna, ma'am, you can speak now.
Still connected to the audio. Why it's not getting connected? Ma'am's audio.
Is it over, sir, or should I take leave, or Aruna madam is saying something?
Yeah, ma'am, you can take a leave. You can take a leave, ma'am.
Okay, thank you, sir. Thank you, sir. Okay.
Thank you, ma'am. Thank you so much.
Madam's mute system with the problem వచ్చింది Haleem Khan. మనం అది స్టార్ట్ చేయాలా? So I don't think it will work now.
No, we don't have any. I mean to say, we already unmuted her. I mean to say, she's co-host now.
Acha, she's co-host, which she can do.
She's co-host. She can speak actually.
Students.
Anyway, shall I ask Harsha to speak for a while? Harsha, are you there?
Sir, good afternoon, sir. Yes, sir. Yes, sir. Good afternoon, sir.
Yeah, you switch on your video.
Yes, sir.
Yeah.
Sir, are you able to see me?
Yeah. You're visible.
Thank you, sir.
Quickly, Harsha. Your remarks, Harsha. What you have experienced?
Sir, I did not understand, sir.
Pardon?
I really thank Department of Botany, Telangana University, sir. And the lecture was really very interesting, sir. This is the first time I understood that there are so many institutes and there is so much of research going on in the field of Ayurveda and traditional medicine, sir. Sir, on a personal note, I have a very small doubt, sir. I don't know whether I can ask it or not.
Yeah.
Hello, sir. So in we have such a huge population of 1.3 billion people, sir, and almost in the month of July, almost 2 million deaths were expected in this country. So is it because of testing, tracing, and treating people, or is it because of traditional food habits or variants like 19A, 19B, or some other genetic reasons why the deaths rate of deaths is very low in this country? So it is very surprising, sir, actually. So,
Vinita ma'am, are you there? Vinita ma'am? Vinita ma'am?
Yes, I am there. Yes.
Our student is asking one question. Harsha, can you repeat the question again?
Yeah, I heard the question.
yes, sir. Yes, sir.
I heard the question. Hello.
Yes,
Hello.
ma'am.
Yes, ma'am.
My opinion, yes, my opinion is that this less death toll in our country may be associated with our Indian food lifestyle, our lifestyle, because it is very different than the lifestyle and food which is taken in foreign countries. So we have Indians have a habit of eating roti, vegetables, cooked vegetables, not raw things in large quantity, and variety of cooked food which is healthy food. So I think this is associated with our lifestyle and food, and not only that, we Indians have a positive mindset of, means most of the Indians are staying in a family, so we have a good family support, so that also has a positive effect on our health. So from social point of view, from diet lifestyle point of view, we Indians are very rich in that thing. So I think this must be the cause why the death rate is less in India.
Thank you very
Thank
much.
you,
And
Harsha.
one more thing, during lockdown, people were eating home food, and that helped, I think, to some extent.
Arnab ma'am, are you able to now speak?
Yes, sir. Sorry,
Yeah.
sir. Sorry for the inconvenience.
Ma'am. No.
Yes. On behalf of Department of Botany, we are thankful to resource persons who have delivered their lecture, like Professor Yogesh sir and Dr. Vinita madam, for their excellent presentation and wonderful presentation. Like we, the department, have invited you all, where we are really benefited, and we are grateful to you. And Professor Yogesh sir has started his lecture right from making us understand for the structure of virus, and he has shown for some images of STEM, that is focused under transmission electron microscope, and he has explained for RT-PCR test and also antigen test. And actually gaining theoretical knowledge is very easy, but gaining practical knowledge is difficult, what I feel. That is, you can get the literature and all students can get the information from any textbook, but really handling the experiment or performing the experiment to get the concluding remarks or conclusion of the experiments performed is really difficult. And the way they have presented the, what is it, their life experiences is really grateful. And Vinita madam has really shared all her experiences and all her work performed, or all her work being done in their hospital. And as student of school days, we have heard that cancer was a dreadful disease. But now slowly we have reached to that statement saying that cancer is curable. And hopefully the citizens of India and all over the world will have that confidence saying that they will be cured even if being attacked by dreadful disease. And seeing to the slides you have focused, madam, really wonderful. And as our head, Haleem Khan sir has told, the statistical data you have shown is really, lively. And it is that which is igniting our minds, saying that even if we are attacked, there's no problem. There is medicine where it can be cured. And Ayurveda is such medicine where we make use of all herbal products. And we as a student or faculty of botany teach to students about economic importance of all such herbs and shrubs. But it is not that where we have that medicine or we have that standardized laboratories where we go for making of this medicine. And you are specialized and expertised in that field. And we are very much thankful to you, madam. And congratulations, madam. And as Haleem Khan sir has rightly pointed out, whenever and wherever we feel like taking your help, and if we need to take your support, we'll be approaching you. And hopefully, we request you to be in support to us and help us under your genuine circumstances. And we are also thankful to Yogesh sir, where he's serving as emeritus professor. Even at this age, he was onto the platform where he has delivered his topic in a manner where all our students would understand and made it more accessible. And overall, I'm also thankful to convener Tarala madam, who is leading this platform, and also congratulate our young and dynamic head Haleem Khan, who is into the department and who's always doing such works, where he's the one who will initiate to write the proposals to science academies. And it's not the first one where he is holding. He has organized as many as such conferences, and we are here to support him. And we also thank our registrar madam, Professor Naseem madam, who is supporting botany department, and hope that all the students of botany department have benefited with these lectures of today, and with the same zeal and with the same energetic motto, kindly see that you join the tomorrow's session. And with these few words, I would like to congratulate the head, and I'm also thankful to my colleague, Professor Vidyavardhini madam, who is with us from morning, and we, the department, are united together where we take help of our other faculty like Dr. V. Jalandhar and Dr. Srinivas. And we also thank the faculty of biotechnology department who are with us and who have shared their experiences. Thank you, and thank you, sir.
Thank you very much, ma'am. Now, Dr. Lata madam from Giriraj Government College. Lata madam, are you there? Unmute yourself, Lata ma'am. Lata madam. Lata madam, can you give your responses for today's lectures? Tirupati sir, are you there?
Sir, good afternoon.
Good afternoon. Please sir, the responses చెప్పండి ఈ రోజు మీరు విన్న లెక్చర్స్ గురించి.
Yeah.
You can
first of all, hearty congratulations, Department of Botany, Head Department of Botany, Dr. Ahmed Abdul Haleem Khan sir, and Professor Vidyavardhini Madam Garu and Aruna Madam Garu, Jalandhar sir and Srinivas sir, and esteemed today's speakers. We are very happy and thankful to you, Madam and sirs. We are very much of enlightened today's session. We hope this is very, very interested and very important. Why? Because this is a very pandemic situation, and this information and very useful and grateful information. We are very much enlightened, and thank you very much for this wonderful opportunity, sir. Thank you, sir.
Thank you. Pooja, our MSc student Pooja. Pooja, unmute yourself and give your responses. Pooja, are you there? Mahendra sir, your experiences. So, from our students, anybody else who want to speak? Hope today's experiences or today's lecture workshop might have helped a lot for one and all. So with this, today's 4 lectures come to an end over here, and hope that tomorrow morning again you will be back and you will be attentive and giving the responses to the lectures by our eminent resource persons. Tomorrow's lectures, we have got 4 speakers. One is Dr. Tarala Nandedkar Madam. She will be speaking on fertilization to placenta formation. And then we have the speaker from National Institute of Reproductive Health, ICMR Mumbai, Dr. Deepak N. Modi. He will be talking about COVID-19 and reproduction, what we have understood so far. And if time spares, he will be speaking us molecular techniques where CRISPR is going to be enlightened by him. And then we have one more doctor, Dr. Rahul Krishnaji Gajbhiye. He's scientist D, again from ICMR and RRH Mumbai. And he will be talking on impact of COVID-19 on pregnant women and their newborns. And we have the one last speaker of day 2, that is Akhil C. Banerjee. He's director of Institute of Advanced Virology, Kerala. He'll be talking on multiple vaccine approaches against COVID-19. So with this, today's sessions and lectures, we wholeheartedly thank on behalf of Department of Botany for keenly attending today's lectures. Thank you one and all, and today the meeting comes to an end. Thank you once again.